All right, now let's talk about Chapter 7, all about membranes. Um, Cellular membranes, plasma membranes, phospholipid bilayers, a number of ways to describe them. But as we saw in the section on macromolecules, we have these types of lipids known as phospholipids, and they are the basic unit of a cell membrane. You have the hydrocarbon tails that stick to the inside of the membrane and are hydrophobic. They don't stick to water. And then you have the phosphate end with the choline as well that sticks to the outside of the membrane, I guess to either side of the membrane, both on the outside of the cell and the inside of the cell, and that's the hydrophilic part. It has a charge, so it will stick to water. And so when you put phospholipids together, they self-assemble into this arrangement in a three-dimensional structure, into a ball-like structure. Excuse me. And when we talk about origins of life, we'll see how that property of theirs helped in formation of first cells. All right, so the cell membrane is not a static, rigid structure. It is flexible, and the phospholipids can move around, and the proteins to a certain extent can move around as well. So we speak of what's called the fluid mosaic model. So there's a fluidity, a lack of rigidity to a cell membrane. And it doesn't just consist of phospholipids, but it also has these proteins embedded in it. And the proteins, as you know, are strings of amino acids in this three-dimensional structure. And the amino acids, and when you have a protein that sort of traverses the length of or the width of the cell membrane, you have amino acids that are in the center that tend to be hydrophobic, and ones on the ends that tend to be hydrophilic. Um, and so again, there's a fluidity to the membrane. The phospholipids can move around, and lateral movement is quite common. The movement from one side to the other is less common, much less common, as you can see, just because it's harder for those the phosphate ends to move through the hydrophobic region of the membrane. But again, this lateral movement occurs quite rapidly. So essentially, in a membrane, those phospholipids are just kind of moving around all the time. All right, so the cell membrane is connected to the extracellular matrix, as we saw in the previous chapter. Just want to talk about a couple things with the extracellular matrix at this point. Of course, its main job is to give support, that is to bind adjacent cells together, and also allow for communication to a certain extent between cells. So these glycoproteins here, that's a protein that's embedded in the membrane that has carbohydrates attached, and that carbohydrate gives them a charge, and so these glycoproteins tend to attract ions and also water and help keep the extracellular matrix hydrated. Um, there are a lot of fibers as part of the ECM that includes collagen. Collagen is a very abundant protein in animal cells, in our cells, or in us, period, you should say. Again, as part of the extracellular matrix, serving a function of support. The amount of collagen varies amongst cells, with some having more and some having less. The cells of the connective tissues have a lot of collagen in them. Uh, Ligaments, tendons, bone, a lot of collagen. Now, there's other fibers as well. There's one called elastin, E-L-A-S-T-I-N, and it gives a certain stretchiness or elasticity to the extracellular matrix. And so that's what, when you just kind of take your hand, your skin, and just kind of push on the skin, and it can sort of move around and stretch a little bit. The elastin is the primary fiber doing that. Um, and there's another one, and I'll go ahead and just name it. I'll spell this out. Fibronectin, fibronectin. And they're being demonstrated right here, and they're ones that connect the cells, the cell membrane, to the collagen fibers, um, and assist somewhat with communication um, in that that intracellular communication. Okay, now these figures over here are just showing there's this technique that's been used to study cell membranes called cell freezing and fracturing. And so what you do is you freeze cell membranes, and you sort of peel the two sides of the membrane apart, the two layers of phospholipids, and you're looking on the inside of that cell membrane, the hydrophobic side. And so what you're seeing, these little bumps, are all the proteins that are embedded in that cell membrane. So you can see this one, this is the inside of that layer. So this is the extracellular layer, the layer that would be sort of towards the outside of the cell, and this is the side that would be towards the inside of the cell, the cytoplasmic layer. And you can see all those proteins that are embedded. That would be these guys right here. They're sort of sticking out when you peel it apart. All right, so the nature of the phospholipids can vary. They can be unsaturated or saturated, and when they are unsaturated, they make the membrane even more flexible and fluid, and when they're saturated, they're less so, more viscous, a little stiffer. The proportions of these unsaturated and saturated fatty acids will vary between animals and even over time within an animal, because animals that live in really cold climates, the book talks about Arctic fish and stuff like that, that tend to have phospholipids with lots of unsaturated fatty acids to keep their membranes from becoming too stiff, where that's not necessary if you don't live in such a cold climate. Cholesterol also is found in the cell membrane, and the cholesterol will helps reduce the fluidity of the membrane, and it also prevents the movement of the phospholipids around. And so it kind of under warmer temperatures, it keeps the cell membrane from being too fluid. And so again, between species and within species, over time, again from the cold season to the warm season, species can adjust the nature of their cell membranes. All right, so the proteins that are in membranes, lots of different jobs. We're going to talk about transport a lot. There are ones that serve as pathways for things to move in and out. Some membrane proteins, they're embedded, they're stuck in the membrane, but they serve an enzymatic function. Signaling, again, signaling from the outside to the inside of cells. Some of these proteins are there to receive these chemical signals from the outside and then transmit that signal to the inside of the cell. Cell-to-cell recognition, some of those glycoproteins that we talked about serve that function. Basically gluing cells together. Talked about that with those tight junctions, I think they were called from the previous chapter. And also some of them are there to attach to the extracellular matrix. So many different jobs for membrane proteins. So membranes have a sidedness, sort of an inside and an outside, and so you can see, as we've mentioned before, portions of membranes can break off, say the ECM, and then fuse with the Golgi, and then others can break off the Golgi and form these vesicles. So it's a very dynamic system, and so you can see that the side of the membrane that's on the inside of the Golgi apparatus and the endoplasmic reticulum and these vesicles becomes the outside of the cell membrane. So those glycoproteins that will stick out of the cell will basically be on the inside of the endomembrane system. All right. So, very short section here. As you probably recall from past studies, membranes, we describe them as selectively permeable. So they're not impermeable, but selectively permeable. Some things can pass through them and some things cannot. For example, in just a layer of phospholipids, when you have water, water cannot pass through because water has a charge and basically will stick to the phospholipids and be repelled by the hydrophobic tails on the inside. So water can't pass through. But it's selective permeability because the cell has the ability to allow certain things to come through and certain things not to come through. And so, for example, we have these transport proteins that allow water to pass through. And these tend to be open all the time. And so water really can just diffuse in and out across cell membranes, but they have to do it through these transport proteins. Other times we're going to use energy and we're going to do active transport to move things across membranes. Some molecules, like that are small and don't have a charge, like oxygen and CO2, these things can readily diffuse across membranes. And so when a cell is doing lots of cellular respiration and using up lots of oxygen will be coming in, and the CO2 given off will be released. And those kinds of things can easily pass across a membrane. Okay. See how much of section 3 we can get done in our video. So again, here's passive transport, and this is something I'm sure you're familiar with. And so it's essentially when objects in space move from where there's more of them to where there's fewer of them. And so if you are in class and, if you happen to be going to a school where there's guys and teenage guys like to wear Axe, and so some guy walks into the room, and it's not long before his Axe odor is diffusing throughout the room, and you can kind of just smell it everywhere because he has put a ton on that morning. So moving from where there's more of it to where there's less of it. So as you can see here, we have this membrane that's selectively permeable. And so there's water on both sides. We've got this molecule that we've added. And it's such that it can easily pass through the membrane. And when you pour it on one side, it will begin diffusing to the other side. And as it starts to diffuse, of course, some are moving back, but when there's more on one side, there's a net flow to the other side until you reach equilibrium, and then they're moving back and forth at equal rates. Now, here's a situation in which we have things moving both ways, and so there are different concentrations on either side, and so the sort of yellowish one is diffusing to the right and the purple one is diffusing to the left. And you can see again there's a net flow of the orange one way and the purple things the other way until again you reach equilibrium and they're both moving across the membrane at equal rates once you've reached equilibrium. So osmosis, we know that as, and I should note up here, passive transport. What's sort of one of the hallmarks of it? No energy required. No energy required. Let's see, I guess I should stop here. I'm just about out of time, so we'll talk about osmosis. We'll start up with that in the next one.